Good morning, ladies and gentlemen. Today, what we are going to discuss is an important topic in the paper called Genomics and Proteomics. The topic is Maldi, M-A-L-D-I. So, what is Maldi? I'll tell you the expansion: matrix assisted laser desorption and ionization. So, this is what the expansion for Maldi. I'm sure this will be asked in the section A. So keep it in mind. Maldi refers to matrix assisted laser desorption and ionization. Right, gentlemen, a small sensitization to everyone. Kindly stick on to the rules and regulations as formulated by World Health Organization and Ministry of Family Health and Welfare, Government of India, to prevent corona. And spread the awareness to everyone and be safe in your home. Right, we'll move on to the topic now. So what is Maldi and why it is used for? So as I told you, the expansion for Maldi is matrix assisted laser desorption and ionization. It is a technique associated with mass spectrometry. Right? So just recall what is mass spec. Mass spec is an analytical device used to determine the molecular mass of the protein. It is used to determine the molecular mass of the protein, and this is how a Maldi plate looks like. This is how the Maldi plate looks like, and you could find lot of small spheres into which the matrix material is embedded. The matrix material is embedded along with the protein to be studied, right? So you have to understand the principle behind Maldi and the application of Maldi. Maldi can be otherwise called as soft ionization procedure. Since we are dealing with most fragile molecule, so-called proteins, it should be ionized in a safe way. Otherwise, we cannot reconstruct the denatured protein molecule and we cannot bring back to its original shape and structure. So to avoid it, we are using a soft ionization procedure. அதாவது புரதங்கள் ரொம்ப மென்மையான மாலிக்யூல் அப்படிங்கிறனால அதை நம்ம என்ன பண்ண முடியாது, ஹீட் பண்ண முடியாது. ஹீட் பண்ணோம்னா என்ன ஆயிரும் அப்படின்னா, the protein becomes completely denatured, அதாவது அதனுடைய ஒரிஜினாலிட்டியை வந்து அந்த புரோட்டீன் லூஸ் பண்ணிடும். ஸோ அதுக்காக நம்ம என்ன பண்ணுறோம், டு எனேபிள் த டைஜஷன் ஆஃப் ப்ரோட்டீன்ஸ், நம்ம இந்த சாஃப்ட் டைஜஷன் ப்ரொசீஜரை இங்கே அப்ளை பண்ணுறோம். ரைட்? ஸோ Maldi is an example for soft ionization of proteins. இதை மறந்துடக்கூடாது. ஸோ நேற்று மாணவர்கள் கேட்டுக்கொண்டதற்கு என்னங்க அப்பப்போ சில வார்த்தைகளை வந்து சில கோட்பாடுகளை தமிழில் சொல்கிறேன். பட் ட்ரை டு ஃபாலோ மீ. ஏதாவது query இருந்துச்சு அப்படின்னா கண்டிப்பாக போஸ்ட் இன் த கமெண்ட்ஸ். வெல், ஸோ the slide what I show you just before is the Maldi plate. We'll move on to the theory session. So it is a soft ionization procedure in mass spectrometry. Right? As I told you, Maldi is an example for soft ionization procedure. It is in connection with MS. Right? Don't split Maldi and MS in a different topics. Right? Maldi is a procedure for ionizing or for digesting the proteins in a simple way. Right? So we can analyze biomolecules and large organic molecules. It's very, very important. Biomolecules, you know, carbohydrates, proteins, lipids, nucleic acid, enzymes, all these molecules are called as biomolecules since they are playing an important role in the biological system, so called in the human body. So they are called as biomolecules. through MS we can analyze the structural details of all the molecules. And what makes the protein to ionize is an important question. Sir, you are telling Maldi is a soft ionization procedure, right? We'll accept. What makes the protein to ionize? The thing is, I'm highlighting the letters, so-called the laser beam. Laser beam alone makes the protein to ionize. So, you may be asked a question. What is the source of ionization in Maldi? You can write laser beam is the source of ionization in Maldi, right? So, Maldi is comprised of two steps. First step is called desorption. And you may ask questions there, what is desorption, right? It is the antonym to the word called adsorption. So, adsorption refers to attachment. Desorption refers to detachment. A simple thing, right? Don't get confused. First, desorption of protein is triggered by the UV laser beam, right? UV laser beam, and the matrix material heavily absorbs UV laser light. See this, the beauty of making the matrix. So matrix, it is a colloidal substance, is coated on the plate, right? In the previous slide. So here we will coat the substance. The substance contain molecules which could trap energy primarily from the laser light, because as I told you, proteins are complex. Structurally they are complex, no doubt in that, but complexity is not proportional to its structural integrity, right? So to avoid maximum disintegration of, maximum destruction of proteins, we use materials to coat on the Maldi plate. Over the embedding material, the protein to be determined are layered. The function of the matrix is to observe maximum energy from the laser light. From the observed energy, part of the energy will be transferred to the layered protein molecules, right? Not the protein molecules directly, not directly observe laser light, because laser will definitely disintegrates the protein into pieces. So to avoid it, we are using an intermediate material called the layer or the matrix. Well, this is the diagrammatic representation of Maldi, right? The plate here refers to, this is the plate, Maldi plate, what I show you in the previous slide. And these spheres are substances used to coat over the surface of Maldi plates. And these differently colored structures are embedded protein molecules, right? This one, this one. So these are all the embedded protein molecules. So after embedding the protein on the surface of matrix, it is subjected to analysis. The first step is ionization. See, from the source of laser gun, heavy intensity of laser light hits on the matrix. See, not on the embedded protein molecules directly. It will falls on the matrix substance. See, laser falls on the matrix. Maximum energy is observed by the matrix substance. So from the matrix, a part of the energy will be transmitted to the embedded protein molecules. So what the laser beam will do? This makes the protein to desorb. So as I told you, desorption is the antonym of adsorption. Adsorption is attachment, desorption is detachment. So here with the laser beam, with the help of laser beam, the embedded protein molecules are desorbed from the matrix, right? This is the first step. And after the procedure of, after the process of desorption, the protein molecules are entered into the next state called ionization, because once they are detached from the matrix, it is in a intermediate state, so that they will be subjected to the next procedure called ionization. So what happens during the ionization procedure? During ionization, the dissolved protein molecules acquire charges, because every molecule wants to be stable. Every molecule wants to be stable in its original state. But what happens during the process of ionization? This makes the protein pretty unstable one because of ionization. So to become stable, it is further degraded, or it is lysed into smaller molecules, right? A very simple mechanism. Question may be asked like this: what is Maldi? Or explain mass spectrometry. Or maybe in 5-mark question, explain the principle of Maldi, like that. But don't leave this topic, it is very essential one. Once the proteins are ionized, the rest of the process are very similar to mass spectrometry. Instrumentation. So what I taught you a couple of days back, mass spectrometry. You can follow the instrumentation of the mass spectrometry in my previous video lecture with respect to mass spectrometry instrumentation, right? The basic principle is desorption. So what is desorption? Detachment of proteins. From where proteins are detached? So as we discussed, proteins are previously coated onto the slide, which is layered with a matrix material, right? So from the adsorbed material, the proteins are detached, right? We'll move on to the next area. First step is desorption, right? Second, the analyte molecules are ionized in a hot plume. So analyte here refers to the sample to be analyzed. Analyte here refers to the sample to be analyzed. Then what is the sample here? The analyte is the protein molecule, right? Don't get confused. Analyte is the protein molecule, right? So this will be ionized. So Maldi is a two-step process. First step is desorption. Second step is ionization. Ionization is mediated by laser light. So this is what you have to remember all through this lecture. Fine. And what are the materials are used as the matrix? There are three different things. 3,5 dimethoxy hydroxycinnamic acid, alpha cyano 4 hydroxycinnamic acid, and 2,5 dihydroxy benzoic acid. Guys, this is very important one. Remember all these things both for your section A as well as section B questions, right? Definitely a question from this layer, from this area will be the most prevalent one in your terminal examinations. What are the materials are used as matrix in mass spectrometry, right? Don't forget to write 3,5 dimethoxy 4 hydroxycinnamic acid, alpha cyano 4 hydroxycinnamic acid, 2,5 dihydroxybenzoic acid. The uniqueness between all these three compounds are the presence of a benzene ring, right? The benzene ring will absorb the maximum energy from the laser light. Part of the absorbed energy will be transferred during the ionization process, right? So this is the principle behind using these organic molecules as the matrix materials. Don't forget. Fine. So these are all the materials what we saw in the last slide. Application. Where do we apply these matrix materials, right? If we use 2,5 dihydroxybenzoic acid, it will be applied in the case of peptides, proteins, lipids, and oligosaccharides, right? In case of 3,5 dimethoxy 4-hydroxycinnamic acid, it is used against peptides, proteins, and glycoproteins. Alpha cyano 4-hydroxycinnamic acid, proteins, peptides, lipids, and oligonucleotides. Since we are dealing with the subject called proteomics, the matrix, the most common matrix for analysis is 2,5 dihydroxybenzoic acid, and the second one is alpha 4-hydroxycinnamic acid. Remember, mechanism of Maldi is done in 3 steps: formation of solid solution, matrix excitation, and analyte ionization. These are all the 3 things. What is formation of solid solution? And this refers to the preparation of matrix. So we discussed there are 3 different materials, and this will be mixed with suitable solvent so that it will become a paste-like material, and this will be coated onto the matrix plate, I mean Maldi plates. Second one, excitation. What happens to matrix to excite? See, all the molecules are in its resting phase in the ground stage level. When it absorbs energy from the laser light in the form of electrons, the molecules are excited. The matrix molecules are excited. From the matrix, a part of the energy will be transmitted to the adsorbed protein molecules so that they are also excited along with matrix. And in the second stage, called analyte ionization, the excited proteins are ionized here, right? We'll see the things in a diagrammatic representation way so that you could understand easily, right? So this is the common way. So this is called Maldi. Here you could find a new term called TOF. Sir, what is TOF? TOF refers to time of flight. T-I-M-E time, O-F flight, F-L-I-G-H-T, right? What is time of flight? So just recall the things what we learned from mass spectrometry. In the magnetic field, the molecules are pretty deflected because you cannot expect there is uniform size of molecules will be produced during ionization procedure. So irrespective of their size, they are deflected. So higher the mass, higher the deflection, lower the mass, lower the deflection. This is what the principle, what we saw in the last lecture, right? So time of flight refers to the time taken by the individual molecules to reach the detector, because detector is the component which will detect the sample, right? So this is the Maldi plate, a virtual Maldi plate, and this is coated with the materials, right, so called the matrix materials. Once they are coated with the material over the matrix, it is loaded with the samples, right? It is given in different colors. See, the color of the sample is given in green, whereas the color of the matrix is given in blue. 2 different form of representation. Once the samples are loaded, it is kept in electrostatic field, right, in which a beam of laser hits the molecules, I mean the sample loaded onto the Maldi plates. As soon as the laser light hits the sample loaded onto the Maldi plate, they will become excited. Excited molecules are carried over to the next phase called magnetic field, where they are deflected based upon their size, and the deflected molecules finally reaches the detector. So we discussed the detector of mass spectrometry is enabled with a structural feature called diodes, which will amplify the signal. Since the signal produced by the ionized electrons are very less. So to amplify it, it is enabled with diodes. Finally, this is how the output looks like, right? Here, the graph 1, MS profile, right? X-axis, it is known for the separation based upon the principle called MS, M by Z. M refers to mass of the protein and Z refers to charge of the protein molecules, right? And the y-axis is known for the intensity and abundance of the molecules, right? How much substance are there in the sample. So while plotting the graph, because everything is a computerized one, our simple duty is to load the protein properly, that's it. The rest of the things will be carried out by the machine itself. We'll obtain the result in the form of graph. To interpret the graph, we should have some basic knowledge in chemistry. This is what important. So a very good chemistry person is required to interpret the result what we obtained during mass spectrometry, because interpretation is very important. What refers to the height of the peak, why this peak is large and why this peak is small, why these peaks are intermediate. So this are the compounds which are separated based upon the mass to charge ratio and its abundance in the sample, right? So don't get confused. Maldi is a very simple procedure. To sum up with, Maldi is a soft ionization procedure used for ionizing the protein sample, and it is used along with mass spectrometry, right? Maldi is not a distinct procedure. Maldi is used along with MS because there are molecules, their structural properties are different. Some are strong, some are fragile. Since proteins are fragile molecules, what we use here is a laser light. Laser is observed by the matrix substance. Observed energy, a part of the observed energy is alone transferred to the coated protein molecules, so that this causes not complete disintegration of proteins. As part of the energy is transferred, the molecules are pretty safe, but they are completely ionized, right? So it's a very useful procedure, not only in the case of proteomic research, but also as you people are the students of genomics and proteomics paper, you should understand the principle of Maldi and why it is applied in proteome research. Thanks for staying with us. Thanks for the opportunity given. We'll meet you in the next lecture. Bye.